Hi, my name is Michael Yu. I'm a PhD student at UC San Diego, and I'd like to first thank the organizers for giving me the presentation, for the opportunity to present this work. And this afternoon, I'm going to talk about work we recently published in Cell Systems about the translation of genotype to phenotype by a hierarchy of cell subsystems. So the problem that we are interested in is, given some training set of genotype-phenotype relations, can we build a model which will predict the phenotypes of new genotypes? Now, now classical approach, which you might dub the network or pathway approach, does this by taking genotypes and mapping the effect of mutations on genes to alterations in some network, some molecular network of, say, gene-gene similarities or interactions. And then alterations in those networks are then mapped onto phenotype. Now, this approach has been very successful in the past years. However, the knowledge in this network is typically represented by pairwise gene interactions. However, we know that the cell is not just, does not just result from pairwise gene interactions, but from the interactions at higher order levels involving more than 2 genes, say at protein complexes, pathways, processes, and organelles. And so in this work, we take that intuition and we consider this hierarchical approach where we take genes and we map them not onto some flat network of pairwise gene interactions, but onto a hierarchy that maps the complexes, processes, and other components of the cell, and then uses that information to map the phenotype. So as hierarchical maps of cell structures, we use two databases, or two sources of information. The first is the gene ontology, which many of you know is this manually curated database that consists of thousands of different terms with millions of annotations to genes across dozens of species. And this is just a global view of the hierarchical structure of the gene ontology. We also considered in this work as an alternative what we call a data-driven ontology, which I'll refer to as a nexo. And this is similar to the gene ontology. However, rather from manual curation, this ontology, the hierarchical structure, is inferred from omics data sets, including PPI and co-expression networks. And this method is automated and it's unbiased. And the way it works is you basically take a network of gene interactions and you hierarchically cluster it into what's known as a directed acyclic graph, or DAG. And so we take this knowledge, this hierarchical knowledge, and we're going to translate genotype to phenotype. In this work, we focused on one particular type of genotype-phenotype relation known as genetic interactions. To understand what those are, let's suppose you have some wild-type cell and you've measured its cell growth. This is just a particular phenotype. And for convenience, we'll normalize the phenotype to a unit of 1. And also suppose we've examined single gene deletion mutants, where say we've deleted gene A or B, and we measured the growth to be 0.7 each. Now, for most pairs of genes, when you delete both of them, what you expect is that the growth is going to be approximately a product of the single mutant growth rate. So in this case, it's just 0.7 times 0.7, or 0.49. So that's what you expect for the vast majority of gene pairs. And that's where no genetic interaction happens. However, in a small set of gene pairs, the growth rate may be slower than expected. That's what we call negative interaction. In the extreme case, it's sometimes known as a synthetic lethality. And in the other side, the growth may be faster than expected. And that's what we call a positive interaction. And so for this work, we took a data set published in Science back in 2010 that measured 5 million pairs of genes in yeast in Saccharomyces cerevisiae and measured if there were genetic interactions. And to our knowledge, this represents one of the largest genotype-phenotype relation data sets out there. And again, the question that we are focusing on is, given a pair of genes, can we predict whether there's, predict its phenotype? In this case, whether there's a genetic interaction or not. So to understand these genetic interactions using a hierarchy, I'll start with some intuition. So suppose we look at the interactions, we might notice that these genetic interactions tend to concentrate or bundle among some genes. In this example, there's many interactions that are crossing between the left group of genes and the right group of genes. And so from a gene-centric point of view, these are just a bunch of interactions across genes. However, if we have knowledge that these genes are actually part of the kinesin and dynactin complexes, then we might be able to reinterpret these interactions, these many interactions, as a single interaction between two terms, the kinesin and dynactin. And further, if we have even higher level knowledge that kinesin and dynactin are parts of microtubules, then we might reinterpret all these interactions as just a single interaction within microtubule-associated complexes. And so if we expand this idea across all interactions, what we find is that there are about 400 and 1,300 of these what we call within and between term enrichments. And these enrichments altogether explain about a little over a third of those genetic interactions in our data, the data set we considered. So taking this intuition, the goal of this work was to build a model that predicts cell growth phenotype in yeast based on how these genetic interactions are hierarchically organized. And more broadly, the more general goal was to build a model that can predict cell function, that is, translation of genotype to phenotype, based on prior knowledge of cell structure, how the cell is composed of components, processes, and pathways. And so this intuition, hierarchical intuition, led us to a computational framework for translating genotype to phenotype, and it works as follows. In this framework, we first consider some genotype. In this example, genes B and D have been deleted. We then take the mutation on these genes and we map them onto some hierarchy, in this case the gene ontology, and we estimate how much has each term in that ontology, each cellular component or process has been perturbed because of mutations in the genes. So in this example, because B and D have been deleted, terms T1, 2, and 3 have been perturbed by one gene deletion, and terms T5 and T7 here have been perturbed by 2 gene deletions. And we can do this across many genotypes and create what we call an ontotype. The ontotype is just simply the set of values we infer across all terms in the ontology. And so in this case, because we're considering double gene deletions, every value in the ontotype is either 0, -1, or -2, representing whether no genes, 1 or 2 genes, have been deleted for that term. And finally, once we have those ontotypes, we then, using supervised machine learning, associate them with a phenotype. In this case, what we use is random forest, which consists of many decision trees that ask logical tests about the states of these terms in the ontotype in order to predict the phenotype. And so zooming back out, the overall procedure is that we take genotypes, transform them into this concept of ontotype, and then use machine learning to associate it to phenotype. And the reason why we call it the ontotype is it's kind of this intermediate feature between genotype and phenotype. Whereas genotype is the state of genes and phenotype is a state of observable traits, ontotypes represents the state of all biological objects in between genes and the broad phenotypes at the end. And so taking this method and applying it on that data set of 5 million measured gene pairs, we find that this method works quite well. In this plot, we see a general correspondence between predicted interaction scores and the experimentally measured interaction scores. These predictions were made in fourfold cross-validation. And also, as a strawman null model, when we randomize the ontologies, the gene ontology or these data-driven ontologies that we used, randomize their structure and plug them back into the model into this framework, we find that the performance degrades substantially, close to 0. So that's our model. The original model is shown here by fgo and fnexo, and the randomized versions are frandomgo and frandomnexo. And also, we compared our method versus non-hierarchical methods, so that using more flat information, and we find that they don't perform as well. We compared against flux balance analysis on a separate metabolic model, and we compared these machine learning algorithms that use molecular networks as directly as features. And lastly, we also examined what happens when you don't use the entire hierarchy, but only, say, use the highest level or the lowest level concepts, say like the lowest level complexes and processes, or the highest level ones. And we find that in either case, when you cut and only use a fraction of the hierarchy, then the performance also degrades. So all these different validations show that the hierarchy is important. The entire hierarchy provides a lot of information about translating genotype to phenotype. And so once we've trained the model, we can visualize what it's done. And so this here shows a global map of what was the logic that was learned to predict genetic interactions. The nodes and the black edges represent what the hierarchical structure of the cell. So each node here represents a component, process, or pathway. And the coloring of the nodes and edges represents a concentration where the model has concentrated genetic interactions. So red here indicates a concentration of negative interactions, and blue represents a concentration of positive interactions. Now this picture is quite hard to see, so I'll zoom in. I'll zoom in into DNA repair. And DNA repair, as can be seen here, is composed of many different sub-processes. And as seen by this pie chart underneath DNA repair, gene pairs underneath DNA repair can either have a negative or a positive interaction. And whether it's negative or positive or simply no interaction depends on which sub-processes those genes are involved in. And so what we can see if we factorize DNA repair, we can see that that there's this red edge between non-homologous end joining and homologous recombination. And this red edge means that the model finds there's an enrichment there's a concentration of negative interactions between these two terms. So in particular, if we delete one gene in non-homologous end joining and delete one gene in homologous recombination, that tends to result in a slower than expected growth. We further validated that this model was correct by going into the laboratory and testing new genetic interactions for 2,500 gene pairs for which both genes affected DNA repair or the nuclear lumen. And what we find is that first, there is a nice correspondence between these two bars here represent the correspondence between previous measurements of those gene pairs and our new measurements of those gene pairs. So there's about a correlation of around 0.6 between these two data sets. However, when we look at, we take our model and model that was trained on the previous data and make new predictions for this new data set, we find that the correlation is actually a little higher. And so this is a little surprising, and we think what's happening is actually that our model, because it is trained on millions of gene pairs, it effectively smooths and understands, it smooths out the noise within any one experiment and identifies what are sort of the overarching biological principles behind those interactions. And just as another example, I won't have time to go further into it, but you can zoom into other portions of this model, for example, to note how the nuclear lumen and its genetic interactions can be hierarchically factorized. Now, lastly, we looked at whether this model can work on more complex genotypes. Note that our model, it was only trained on double gene deletions. And so we wondered whether it could predict the phenotypes of genotypes where more than 2 genes were deleted. And to investigate this property, we looked at this study in 2014 where they screened double and triple deletion mutants. And in the study, they deleted the ASF1 or CAC1 gene in yeast, and they also deleted another gene, which may have included a gene in the HER complex. So there's HER1, 2, 3, and HBC2. And what they found in the study is that double mutants, where you delete CAC1 and one of the genes in this HER complex, that resulted in a slower than expected growth rate, or a negative interaction. However, when you delete one more gene, ASF1, so you delete 3 genes simultaneously, this negative interaction was suppressed. It grows as expected. These were the experimental findings, and when we took our model that was trained only on double gene deletions and made predictions from it, we find that it recapitulated both the double mutant and the triple mutant phenotypes. And examining further how this model worked, we took a look at what functions were these genes involved in. And so ASF1, CAC1, and genes in the HER complex, they are involved in chromatin organization, nucleosome organization, and replication-independent nucleosome assembly. And we also looked, based on this hierarchical organization, what was the decision logic involving these terms to predict the correct phenotypes. And so in summary, what we've been able to do is predict genetic interactions accurately using a hierarchical model of the cell. This model could be based on the gene ontology or a data-driven ontology like Nexo. And we find that it outperforms non-hierarchical methods. It can be we further validated in DNA repair and nuclear lumen, and it has the potential to generalize to higher order genotypes. And a little bit of modeling intuition of why this worked. We think that because we use the hierarchy, this hierarchy spans multiple scales, and it allows the model to examine and finally select what is the right scale that explains each genotype-phenotype relation. For some genotypes, you may need to look at a low level. For others, you may need to look at a higher level. And it's important to consider all scales simultaneously when training a model. And lastly, we think that this work demonstrates that modeling in the cell generally can be divided into 3 stages. The first is mapping cell structure from physical information, say from omics data microscopy. The second is mapping cell function from genetic information. And finally, integrating those two together through these gene ontologies and ontotypes. If you'd like more information, you can take a look at our publication in Cell Systems. We had a cover on it with a psychedelic image showing mutations in the genome and how they get funneled into this hierarchy of components and processes within the cell, finally producing this swirly image of phenotypes. And I'd like to end by thanking the many people who are involved in this project, notably Michael Kramer, Janusz Dukowski, Jensu Ma, my PhD advisor, Trey Edeker at UC San Diego, and also our collaborators, included Roded Sharon. Thank you.